So this time, I will talk about the microfluidic bioprinting of heterogeneous 3D tissue constructs using low-viscosity Bioink. So what is bioprinting? Bioprinting is one of the 3D printing techniques, but which is using the polymers and also the cells, and which is applied for the medicine mainly. But there is an issue that the composition of printing material has not been fully optimized and characterized. So this author, Professor Ali, has been working on the bioprinting for the long years, and he has been working on also biomaps, biomaterials, and tissue engineering, and so on. And the JELMA is one of the main materials he has been used, and the challenge in JELMA printing is low viscosity. So the low viscosity helps the spontaneous organization of specific stem cells, but sometimes they lead to the low printing quality, so there is a mismatch in the JELMA printing. So at this time, he used the alginate, which is structurally stable both before and after curing, and also which is fast curable by chemical reagents. So he actually integrated JELMA and alginate in the same Bioink, and he used this bioprinter to deposit those bioinks. So here is the overview and what is the novelty in this research. One is the creation of viable 3D cell lading constructs. And the second one is spreading and migration of embedded cells, as you can see in the pink color. And the third one is positing of additional cell types. And the fourth, lastly, they actually validated the simultaneous multi-material deposition. So let's talk about the first one, the creation of 3D cell lading constructs. So they actually fabricated this kind of specific nozzles, and the outer nozzle dispensed the curing reagent, and the center nozzle dispensed the JELMA and the alginate at the same time. So as soon as they dispensed, the curing reagents bind with the alginate fiber, and actually this Bioink is spontaneously cured. So what's happening in the alginate, as you can see in the right bottom, and let's move on to the left bottom. They actually characterize the ratio of alginate to the curing reagent, and also they check the shear rate versus viscosity. So with alginate, the JELMA can stay in the almost same viscosity. So this shows that the alginate helps the JELMA stability. And after the bioinks are deposited, the JELMA inside is cured by UV, as you can see on the right top, and so the bottom left figure shows that the UV exposure until the 30 minutes is okay, and let's move on to the right. So the cellular viability remains almost same, but actually the bottom part is slightly affected by the lower, but actually the viability remains almost same. Secondly, they check the spreading and migration of embedded cells. They use the F-actin and the CD31. So actin is housekeeping gene, and they actually confirm that the transcription, as you can see on the bottom left. And when it comes to CD31, which is the membrane protein highly expressed on the endothelial cells, and which is good for the checking cellular migration. So as you can see on the bottom right, so the pink one is the CD31 cells, CD31 proteins, and yeah, those proteins are well distributed, and they actually by using those F-actin and CD31, they check the spreading and migration of the cells. And they use the cardiomyocytes for the contraction testing. So they simply put the cardiomyocytes to the bioprinted networks. And as you can see in the graphics, the contraction of cardiomyocytes are regularly observed, and the scaffold may help the spreading and migration of cardiomyocytes, and also the sustainable mechanical property. And lastly, they validated the simultaneous multi-material depositions. So this system allows the rapid printing of bioconstructs formed by different materials. So as you can see in the red and green colors, and that may help replicate the heterogeneity of the native tissues. So this doesn't show us any applications, but this would be the important fabrication technique in the future. So they actually validated the printability of the Geoma alginate integrated Bioink, and also checked the spreading and migration of cells. And also the post-seeding is valid, and also they developed the simultaneous deposition system. So 4D printing has been really popular for the past 5 years. And this study actually characterized the UV exposure conditions. So this would help and contribute to the further development of light-driven 4D biomaterials. So I actually want to use shape memory polymers in this kind of bioprinting. And the nozzle customization will be really important. As you can see, this is developing the perfusible vascular constructs. And they actually articulately fabricated the nozzles. So this will be one of the important fabrication techniques in the near future in the field of bioprinting. So our group has strengths in the 2D printing, but actually we want to integrate the 3D printing technique to this kind of 2D printing technique. So yeah, also the shape memory polymer will be really important for us for the 4D and the further development of the functional materials. So thanks for listening, and feel free to ask